Hey, my name is Kelsey West, and my background is in immunology and really genetics, focusing on bioinformatics science. And I am currently a data science program or project manager at a nonprofit. And so today I get to talk to you about methods and protocols and how accurate and well-documented methods are essential for transparency and collaboration, as well as building trust in science. And I'll introduce some tools that will help us document and share protocols effectively, with a special focus today on protocols.io. So reproducibility is really at the heart of good science. And I know Cameron was really um, preaching this earlier as well, but if a method can't be reliably repeated, its findings become seriously questionable. And unfortunately, almost every area of research is finding this problem, and their findings aren't reproducible. So I've highlighted here on the right a study that was looking at findings from the field of psychology and the efforts of trying to reproduce about 100 different findings from the field. And they found that only 39% of those were actually able to replicate what they had found in their original study. And this isn't just limited to social science. This is also issue in life sciences. So in cancer biology, a group of researchers looked at over 193 experiments from 50 plus papers, and they found that 2% of those experiments had access to open data. So only 2% allowed you to look at the data associated with that experiment. And 0% of the protocols from all of those experiments were completely described. And I'm with Cameron, he mentioned this earlier, but as a researcher, there's nothing more frustrating than getting excited about some kind of method or experimental procedure or an evaluation or assessment you saw someone do, and you go to look at their materials and methods to find that they have little to no detail on how that experiment was conducted. And that means that you can't reproduce their findings, but also you can't apply it to your own work as well. Even the topic, I've seen people in the chat mention machine learning, and it's the hottest topic on the block right now, but it's not immune to the reproducibility crisis. And researchers from Princeton have been looking into the different models that have been built and issues with data leakage and modeling errors in the papers that then continue to build on other models that are published actually have led to effects on 648 different publications. And as researchers, we really rely on the clear documentation of methods and these instructions and protocols and manuals that we create. And many of the traditional documentation, you might feel like this astronaut lost in space of all the different lab notebooks that are handwritten or scattered digital files that are on your computer at your lab or your office or on your personal computer. Or maybe you're filled with sticky notes, as my previous professor liked to do. I don't know how many times I've walked into professors' offices with just stacks of yellow paper notebooks, just filled with notes and different protocols and methods that I'm not sure if they ever go back to. But all of this creates scattered resources, and it really causes effects in trying to reproduce any of these research processes. So all of these challenges in traditional documentation can lead to missing details, vague instructions, and really slows down our progress and ultimately wastes resources and creates unnecessary confusion. And to address these challenges, we really need structured, but also digital solutions that can help support the transparency and collaboration to really build on those open science principles. And I like to think of it as building a solid foundation for your research, but also all the other research that builds on your work and the research communities in the future. And the benefits for sharing open and detailed protocols are going along with those FAIR principles that Cameron had mentioned before. So making sure everything is findable, available, accessible, and interoperable, as well as reusable. So really focusing on that reusable side to this. And it really matters because, as I mentioned before, we're reducing cost and time and really truly fostering trust in the science and the research community. When you use accurate and clearly defined methods and instructions, This not only benefits you in your future research and be able to go back to your own work, but also for your other lab mates, other researchers in your institution, as well as the scientific community as a whole. You can promote transparency and also really gain recognition and credit for your work by publishing your protocols and methods separate from the entire publication. So you don't need to just wait until that entire publication is ready for publishing. You can also share this more easily and collaborate across different fields with sharing these methods with your peers before publication and allows for collaborative improvements across labs and institutions. There is really your methods for or documenting your methods really need to be clear and simple, but making sure that you include your necessary reagents, instruments that you use, conditions, and any kind of evaluations or assessments that all go into the protocols you're developing. And a lot of these points I have shown here on this slide are required by many funding agencies when actually submitting grants or funding. So it's even more crucial to follow these steps to staying compliant with the industry and institutional guidelines. Unfortunately for us, there are several digital tools available that help us standardize and share protocols. Now, this is purpose of just sharing the tools that are available and what's out there. This is by no means a comprehensive list, so please share in the chat if you're aware of any others. These are just a few I've come across in my time that I wish I was aware of a lot sooner. So one of these is called protocols.io, and it is an open access platform that allows researchers to create and modify as well as share detailed step-by-step protocols. It allows you to track with version control as well as using DOIs for assignments. So like Cameron mentioned, getting that citable aspect for that digital object identifier, as well as a lot of collaboration features where you can share entire protocols with lab members or a bigger institution, depending on your needs. There's also BioProtocol, which is another platform that allows researchers to formally publish protocols and ensure that they are peer-reviewed, as well as accessible to the broader scientific community. Benchling is a little bit of a different type of tool as an electronic lab notebook, so also known as an ELN. There are a lot of different services out there for ELNs, but this really ensures that you can manage experiments and protocols while sharing collaboratively with your team with that cloud-based electronic lab notebook. There's also Open Lab Notebook and Open Wetware, which are groups of communities of scientists that have shared their transparent lab notes and protocols to promote real-time sharing of these methods that they're developing. And as Cameron mentioned before, and he's going to go into detail further later in this training, there's also GitHub and GitLab. And while these are mainly thought for software development, these platforms allow for version control for protocols and scripts and analysis pipelines. But the caveat is, just like Cameron mentioned, is that these repositories can ultimately get deleted at some point. And while the other options listed above provide a method of getting a DOI, so it really allows you to ensure traceability and have those identifiable methods more so than accidentally deleting a repository and losing those methods for anyone to go track down. It would be kind of impossible to find. And each of these tools serves different needs, and depending on your workflow, I encourage you to search all of them, and even searching these, I'm sure you'll find more tools out there available. So really just look into the one that fits your type of workflow and process. So we're going to go into protocols.io. I'm just going to highlight this one today. I am no means by sponsored or we're told to present any of these specifically. Protocols.io, I just have personal experience with, so I wanted to highlight that today. And really, they have several different core features that I'll be going over, which is creating a protocol. So you can do that step-by-step process that the interface walks you through creating those, adding your necessary reagents and materials. It also allows for that DOI assignment. So that important question we always get, how am I going to get credit for my work? And assigning a DOI to your methods and protocols is one way to make that happen. It also has a lot of interactive features and really dynamic, which allows you to get feedback from the community as well as sharing workspaces, which will store all of your protocols that you can share with your peers, lab members, different departments or institutions. And furthermore, the couple of my other favorite features of this is that you can add images and videos directly to your protocols. So I'm one of the first people to go on YouTube and figure out what something is actually supposed to look like. Is it supposed to be turning that color? Should I have, shaken that reagent longer or something like that? And this allows you to append those images and videos of your protocol right with the protocol. So that information is not lost. It also allows the option to submit your protocol for peer review. And you can get this through multiple different journals that they allow you to select from, further increasing that citability and your references for your future. So I'm going to go in, and this is a screenshot of the protocols.io homepage, but I am going to share the live screen, hopefully. Here we go. And so this is what the main screen of protocols.io looks like. And you can create an account for free. I'll mention that at the end of this talk and show you a little bit about that. But when you get to this main page, we're going to have a search bar. And so this search bar, I would encourage you, even if you decide not to use this to store and share your protocols on, you can come here and search for any kind of protocols for free, no sign up even needed. And you can search for protocols in any kind of field. So this isn't limited to just life science. This has things for software engineering, electronics, soil sciences, archaeology, different types of science and space groups as well. So there's over 12,000 protocols on this platform as of the last notes that I looked at. But I would encourage you just to search here. If you need a more detailed method, maybe the publication you're looking at isn't giving it to you. Maybe someone else had published something similar already on protocols.io. So please come on here and search. My background being bioinformatics and genetics, we're going to actually search for something around RNA. So we're going to type in RNA in this search box. And we can see RNA is a pretty broad blanket term. So there's probably a lot of protocols out here on this site looking at RNA, and you can see there's over 1,700 results that popped up. So each of these is a protocol that someone has published on protocols.io. They also, with the blue icon, you can see that further, it can be a publication. So the ones with the blue icon have represented these individuals have published this protocol as well. You can see the different workspaces they're a part of. And I do want to highlight that. So we'll take, for example, here, this protocol for RNA precipitation by Liz Hughes. And she is from the University of Edinburgh. You can see how many views she's had on this protocol. So over 17,000, which is pretty awesome. And she's a part of a workspace. And this workspace is called Wallace Lab for Fungal RNA. And what's cool about workspaces is that while Liz is part of this workspace, you can actually go and follow certain workspaces. So just like you would follow on social media, you can follow a particular lab if they have a workspace, or a topic if there's a particular workspace that's in a larger group of a topic that you're interested in. So then you can be alerted for all the protocols and the changes that are followed up with in that particular workspace. So we're going to go ahead and click on this RNA precipitation protocol just for an example of what a simple protocol on this website would look like. But first of all, you get the title, but then you get this DOI. So this is what I was talking about, what's important for citability. So we want to be able to give Liz credit for her work, and she's able to do that so we can cite that DOI. Also, it gives you more features in the steps. So the basic information for a protocol is the steps on how to conduct this particular method. And hers is very simple, only 7 steps, which is great. But some of these are more detailed or extensive depending on what type of method you're looking at, right? That can go on for quite a bit longer. But they have different metadata fields here that you can add additional information for, like guidelines. She recommends certain environment that you should conduct this in, certain type of lab equipment you might need. as well as metadata. And metadata is an important feature on here because it gives you options for PDF as well as HTML and the citation for this particular protocol. What I also like about this is that it provides a QR code that will directly link you to this protocol. So maybe for your next poster presentation or presentation in general, you can include the QR code instead of taking up that space on your poster and increase readability and interactiveness with your audience. So if we come back up here to the different features, we can also look at the materials. So just like a recipe, when you're making some kind of new food, you're going to need all of these ingredients, all these reagents to make this protocol happen. So maybe collect those before you run around the lab and running around with your head cut off, or so that you don't miss any part of this protocol. And then the metrics, we can look at how many views this protocol has had over time. And like I mentioned, she's had over 17,000 views, which is really impressive for this particular protocol. 9 people have bookmarked this, so you can also bookmark a protocol in your own workspace to make sure that you can save this and then access it as quickly as possible from your favorites. And similar to GitHub, we have this option also of copying and forking a protocol. So this is similar, as I said, to GitHub, where you can copy this protocol and modify it and share it within your own workspace, but it preserves the link to Liz's protocol to get the original for crediting. So making sure that we're keeping that link associated with the protocol so Liz gets credit, but then also you're getting credit for the modifications that would be adjusted if there's anything you need or find works better for your particular situation. Maybe you apply this protocol not to fungal RNA, but you apply it to bacterial RNA or something of that nature. But it always keeps the original link to this protocol for transparency and identifiable factor. So that's how you would search for a protocol, an example of what one would look like on this website. So we're also going to create a protocol. So another core function here at Protocol IO. And so on this left-hand menu, we can again search for any protocols that we want to do, but we can also create a new protocol. You can also create a new workspace. So that's where you can share all your files with different team members, lab mates, things like that. Mine here is called Kelsey West, which is my name. And so we're going to click this button here to create new protocol. And it's going to come up with a couple options. A new feature they have added is that you actually can import a protocol from a PDF. So if you have a PDF version of a method or protocol that you want to get added and save yourself some typing, some formatting steps, you can import it via PDF. I will say to make sure you double check and even triple check to make sure your steps are imported properly and the concentrations or times were inputted properly into this platform. We're going to select to store it in a certain folder, any kind of folder that you have on here, and hit continue. And then your first use of this, it will give you a step-by-step guideline on how to use the protocol.io setup. And we're actually going to skip this guide for the purpose of this talk, but encourage you to go on there and do the tutorial on your own. I like to think of this as a Google Docs kind of setup. So we're going to create a protocol called RNA purification and set that up here at the title. You can insert different sections as well as steps. So sections I like to think of as broader headings and different sections that you might have, maybe your experiment or your assessment, evaluation, whatever you're trying to input as a protocol has bigger sections involved. So maybe you want to create multiple sections and not just different steps. So we're going to name our first section collecting RNA. And our first step would be to collect, actually, we're going to collect cells from a tissue culture dish. This is all just an example from an 80% confluent cells. That just means that 80% of the cells are covering the entire plate that you're collecting from. We want to remove the media from the TC dish. And then the next step we're going to add, so we're going to add a new step, we're going to add something. And this leads into my next component feature that I want to highlight. Over here on the right side, we have components. So protocols.io has already done some of the more specifics around the features that you would add to a protocol. So it has all these different specifics that you can add that will add clarity and transparency to your protocol. So I'm going to add an amount. So I want to say I'm adding 1 milliliter of Trizol. Now, some of you may know what Trizol is. Most people might not. So if someone was coming to read this protocol and utilize it in their research, they're saying, what's Trizol? I don't know how to get Trizol. I don't know where to buy it from. What is the catalog number? We can come over here to these components again and click reagent. And by typing in Trizol on the reagent list, it'll generate list of available Trizols. So all the Trizols that are formulated maybe by different companies or specifics to your lab or things like that. You can add a custom field or you can search through this drop-down method. But adding transparency to what specific reagent you used and not just using generic terms adds to the accurate and clarity for your protocol. So we're going to say we use this Trizol reagent from Thermo Fisher. So anyone who comes here and looks at this protocol says, okay, she added 1 milliliter of this Trizol reagent. So I need to get that Trizol reagent if I want to replicate this. So you can add multiple steps, but for the purpose of time here, we'll say that's our ending step for this protocol. But there are so many different components on here. It doesn't just end with lab bench or wet bench, wet lab components. We have different samples, we have different steps, protocols, even command or citations, data sets, any kind of software you might have used for an analysis protocol, geo coordinates for all of my geospatial people I saw in the chat, safety information, a lot of different components that you can add on here. And what I really like about this is that when you go to view this, so we're going to view this protocol as an example, and this is an example of what it would look like in protocols.io. When you add those components, it really highlights for the user the specifics of this protocol and what are the details of it. For your use, I like it because it also highlights maybe I didn't have this accurately. Maybe it's 500 microliters and not 1 ml. And I could say, oh, that's not right. That instantly highlights for me that that is a specific I need to change or modify, and it might not be correct. And for someone using this protocol, it can also highlight for them what are the core factors that I need to pay attention to when using this protocol to reproduce what this person has done. So I really like those different components for those features. And when you go to materials, every time you add a reagent, it's automatically going to add it to your protocol materials so that those are there and listed and someone can gather those before they try to reproduce your protocol. So if we go back to the edit function in this protocol, we can also add description. The images and videos I was talking about, you can add here. You can add the guidelines like Liz Hughes in the RNA precipitation protocol we looked at. You can add references, so maybe you reference other protocols in your protocol. So making sure you accurately reference those here. The materials, as I mentioned before, if there's any additional ones, as well as acknowledgements. And with this, while you're making this protocol, you can also share with other researchers. So a person does not need a protocol.io account for you to share this with them. But if you want to collaborate on a protocol, you can share this protocol with them so they can add their own comments, maybe make adjustments. So doing that amongst the lab, amongst a small group of researchers or collaborators is really efficient in making sure that everything is clearly documented and saved in the protocol. No step is missed, so someone could reproduce your work. So once you're once you're complete with finishing out your protocol, you have the options to publish and reserve a DOI. So again, that's really critical for making sure that you get your credit for your work and that it's citable. You can also submit the protocol once it's published on protocols.io, you can submit it for peer review. further enhancing your citability as well as the reference you can get and keep gaining references for your future as individuals use your protocols. So with that, that's the generation and how you can start looking at creating protocols on Protocols.io. They have so many webinars and so many trainings. I would highly encourage you to look into those trainings. And one of my favorite features with Protocols.io is the ability to cite Protocols.io as your materials and methods. So I don't know about you, but for me, every time I would write a new publication using the similar materials and methods I'd used, trying to reword or not plagiarize myself in rewording those type of materials and methods gets kind of exhausting, and it is a lot of time spent. So this is a simple way you can cite your Protocols.io workspace that will contain all the protocols for a certain publication or a certain set of work, and anyone can access those. This, again, you can search protocols.io for any protocols that are available, and you don't need specific access for any of those methods that are published on there. So I love this feature and keeps it simple and directs people to your whole line of work that have step-by-step processes and clear methods. Another thing I want to highlight is that protocols.io actually has protocols for using their site. So here on the left, we have getting started. So that will show you how to log in, how to set up a workspace and get started on protocols.io. We're going to share those links in the chat. And then we also have examples of different protocols from different fields. So I promise this is not just life sciences on protocols.io. This is social sciences, different engineering groups, electronics. The list goes on. Like I mentioned, there's over 12,000 available on this site. So please go on here, get inspired to publish your protocols and methods. And with that, this is really a call to action for users to really look at all those different tools that I mentioned and find the one that best suits your needs. You know, maybe protocols.io isn't for you. That's perfectly fine. But I would encourage you to go look at the other tools as well as do your own research and look for additional methods as well. So we can really build the foundation of open science with these reproducible materials and methods and really contribute to the community to enhance open science, but enhance the research community as a whole.